when you switch textbook editions, or just textbooks in general, it's like I thought I updated all my PowerPoints to everything, and then nope, there's always like one page number I forgot to update, or one chapter number on one slide somewhere that I forgot to update. But mini unit on genetics, and so sometimes we mix this around where it goes into the lecture, but it kind of just fits somewhere in here before we start getting into bacteria and some of our other units. So microbial genetics, some stuff is going to be reviewed. We're going to review just DNA structures, but we're also going to compare it, eukaryotic DNA versus prokaryotic DNA, go over those wonderful transcription, translation processes, talk about mutations. This is probably all my next slide on what the PowerPoint is all in here. Mutations. And then also talk about how DNA can get from one cell to another. That conjugation pili is not the only way the DNA can go from one cell to another. So we'll see how far we get today. We'll finish this up on Wednesday. So just a little bit about first, what are we talking about when we're talking about genetics? So genetics is the study of inheritable traits. You can inherit them. I don't know if this is a good black one. As expressed by the genetic code. Meaning your genetic code, your DNA, your sequence of A's, T's, C's, and G's, they control various genes. Those genes can get passed on, meaning they're inheritable, and we have all these different features about us. Now, when we talk about someone or something's genome, that's the entire genetic makeup of an organism, meaning we know the DNA, every code, every strand of DNA, every gene, everything that makes up a particular organism. We know it all. And it wasn't that long ago, early 2000s, that we finally finished getting the whole genome of humans. That we know every genetic code for that makes literally like everything in every cell that controls everything. We finally figured out the genetic makeup. So what a genome includes, if it's the entire genetic makeup, it's all the genes, and a gene is a little snippet of DNA that codes for something. I mean, you've got a gene that controls your hair color, you've got a gene that controls your eye color, you've got a gene that controls like everything. And the nucleotide sequences, meaning your A's, T's, C's, and G's. What is their order in your DNA? So that's the entire genome. It's not just knowing A's, T's, C's, and G's, it's also knowing each little gene and what it codes for. Let's see if my clicker works. So since we are going to talk about DNA, and I know this is not the first time you've ever heard of DNA before, because even my kids in like middle school age are learning DNA. So a little bit of review, things you probably already know about DNA. One, it's double-stranded. So again, these are just things up here that is like, things that make sure you remember. DNA is double-stranded. It's in a helix shape, which means it's got that twist to it like the old telephone cords. It's made up of nucleotides, and there's 4 different nucleotides. There's the A's, T's, C's, and G's, your adenine, thymine, cytosine, and guanine. So there's 4 different nucleotides it's made up of. DNA is also anti-parallel, meaning the 2 strands, because they are double-stranded, run in opposite directions of each other. And so you'll see that even on this little picture where this side of the strand ends in a 5, but the other one ends in a 3. And if we followed it all the way around, if this one is a 3, the other side is a 5. If this one's a 5, the other side is a 3. The 3 and the 5 have to do with just the structure that ends up at the end, but they run in opposite ends of each other, because that's going to come important today when we talk about making of new DNA. The fact that they run opposite directions becomes an issue when we start to make new strands of each, meaning that making of new DNA also has to run in opposite directions because the strands are in opposite directions. In the strand, so they're the anti-parallel, they're double-stranded. That outer part of the strand is where we find the phosphates and the sugars. The sugar, that little pentagon, the sugar in DNA is deoxyribose, which is where it gets its name. It's deoxyribose nucleic acid, for DNA. So you have your phosphate, you have your sugar. It's called the phosphate-sugar backbone. It's that outer edge. And it's only the inside part where the two strands come together where we find those nucleotide bases. And how they pair up in DNA. Always A pairs with a T. Spells at. A always pairs with T, and C always pairs with G. I always think that they look almost the same, a C and a G. The G just has a little extra line to it. Anything else DNA-wise I want to go over? I don't think so. Oh. Now, looking at different DNA structure in prokaryotes versus DNA structure in eukaryotes. So the prokaryotic genome, so this are any kind of prokaryotic cells, the very simple cells, bacteria are prokaryotic cells. Their genome, meaning their entire DNA and all its genes, contain a single chromosome and then all the proteins and RNA that hold it together. And that chromosome is haploid, which means it only has one copy. Because we have 23 pairs of chromosomes, because everything's paired up. Prokaryotic cells are one copy, that's it. So I always think haploid sounds like haploid. They have half the number, like we do. Generally, their chromosome, their DNA is in a circle. Ours are not. And because they don't have, prokaryotic cells are too simple, they don't have a nucleus, that circular DNA is just found in what's known as the nucleoid region, which means it's kind of like nucleish, but it doesn't have any kind of membrane. It's not structured like a nucleus. Now, this is just to show you a big old bacteria cell. Here's that single circular chromosome. And yes, if you take a long enough circle, it does start to twist around on itself, but if you followed it through, it's still one continuous circle of DNA. But bacteria and prokaryotic cells have something a little more unique too. They can, not all, but they might have these little tiny plasmids, these little snippets of DNA. They replicate all by themselves. They're like their own individual unique DNA. They're not essential for the bacterial for survival. They don't have information on them that codes for replication or anything else. But they can have some survival advantages. And one of the biggest one is because it's DNA, it might have genes, it might have little snippets of information that code for something. The information that could be on these plasmids might be information that causes them to be resistant to different antibiotics. And we saw that in that video, it was a plasmid that had that NDM1 gene. A gene is just a little snippet of DNA that coded for that bacteria to now be resistant to antibiotics. And these plasmids can easily jump from one bacteria cell to another bacterial cell. So they're not necessary for that bacteria to live, to do its normal stuff, but they could give it some serious advantages. Now, eukaryotic genomes, we are eukaryotes, there's generally more than one chromosome per cell. Again, we have 23 pairs of chromosomes, or 46 total. The chromosomes are diploid, which means they are always paired up. They're linear. If I had to draw chromosomes, you're generally just going to draw a big old X. They're linear, they're not circular. And eukaryotic cells have a nucleus, and that's where you're going to find the DNA. Cells carry plasmids, those little snippets of DNA. But there are some fungi and there are some protozoa that are still eukaryotic organisms that do carry plasmids. We don't, but some eukaryotes can carry plasmids as well. Now, this is showing how you can take DNA and wrap it around and wrap it around and wrap it around and still fit it into a cell. So DNA is extremely skinny, like extremely skinny. In just one cell in your body, in the nucleus of that cell, when you guys saw your nucleus when we scraped our teeth, you saw the nucleus, you magnified it 1,000 times, so it's still small. The DNA that fits inside that nucleus is about 6 feet long, like that's taller than I am. So it's very, very skinny. So it has to fit inside that little tiny nucleus. And so it wraps around these little things called nucleosomes. And so the DNA is the purple. It wraps around those. They start to wrap around themselves. This whole thing starts to wrap around. It's like constantly wrapping and twisting and coiling until you get a super, super tight chromosome to all fit into one little tiny nucleus. Now, when we need more DNA, because anytime a cell undergoes mitosis, anytime we start to make new cells, obviously we need new DNA for those cells as well. So to make more DNA, the big thing you need, and like if we're making something, it's an anabolic process, because anabolism is building things up. The catabolism is breaking them down. So to make or build anything, you need two main things. You need monomers, that's your building blocks. If I'm going to make a building, I need bricks. And you need energy, because the bricks just don't assemble themselves. So you need the basic building blocks, and you need the energy to put it together. Your basic building blocks is you need that sugar, phosphate, and the nucleotide. You need the backbone, you need the nucleotide base itself. But what's nice is that when we're building these different DNA structures, we do have a structure called a triphosphate deoxyribonucleotide, which is really long. It's your nucleotide. It's still going to have your sugar, your base. It's going to have your phosphate group, but instead of just having one phosphate on it, it has 3 of them, hence the triphosphate. And just like ATP, adenosine triphosphate, there is energy between those phosphate groups. And so by having this called this triphosphate deoxyribonucleotide, is you have your nucleotide, and you now have energy that goes with it, because you can break off a phosphate group, just like ATP, and you now have the energy to start assembling all of these new DNA structures and putting it together. It's great. We've got one thing that provides us both that monomer, the building block, and it provides us the energy to put it all together. Now, when we make new DNA, there's always going to be one original strand and one new strand. And this process of having an original strand and a new strand is what's known as semi-conservative. To eventually figure out that when new DNA is made, that DNA is ripped apart, and one of the original strands is found in the new strand, plus like a brand new one. That there's always an original plus a new one. A long time we had no idea, again, until we had to confirm all of this. We didn't know if this was the original DNA strand. Oh, I don't want a chromosome looking. If this was the original strand of DNA, we're like, well, maybe it still keeps the original strand, both of it, but it makes a copy of both, and now you've got 2 copies, but one of them is completely new and original. We had no idea. But eventually we figured out that no, it does get actually yanked apart. And then one of those original strands stays until we get 2 new ones. And it took quite a bit of testing just to figure that out all in itself. Now, this is to show you, again, you have your original double-stranded helix, but we need to make new DNA. And so it's kind of like a 3-step process. From like my simplified version, I mean, I could talk about this for 2 lectures, and I'm not. Simplified process. 1, we have to yank that DNA apart from each other. Is held together. Now I'm going to go back a second. These bases are holding it together, your A's and T's and C's and G's. Luckily, the A's and T's and C's, the G's that hold the 2 strands together are held together by a pretty weak bond, so it's not that hard to yank them apart. So we first have to yank apart the 2 strands, and there's an enzyme that does that really well. It's called DNA helicase. It just unzips the two strands, it pulls those bases apart. Then we have to add some stabilizing proteins, because if something was in that helical shape, it's going to want to go back into that helical shape, and we want it to stay straight while we go through the DNA process. It's the same thing. If you tried to straighten out any type of cord, it's going to want to just, coil right back up on itself. So we add some proteins, and then we add another enzyme, enzyme ACE, called DNA polymerase. And that's the DNA polymerase's job, is to bring in the new bases. If there was a T, it would bring in an A. If there was a C, it would bring in a G, and so on. So it's like one enzyme unzip, proteins stabilize it, the polymerase adds the new bases. You're like, that's super simple. The basics. Because DNA polymerase can only add bases on the 3' side. So if I'm going to want more colors. Polymerase can only add bases onto a 3' side. And so if it's unzipping going this direction, we have to, because we're unzipping that going to that direction, I'm going to go a different color. This is a new strand, because this is the 3', this now has to be the 5, and this has to be the 3'. And so as it unzips, it just keeps adding bases, one after the other, always on that 3' side. Not an issue. You unzip to the left, you just keep adding on to the left. The more you unzip, the more you add. But we run into a problem with this other strand, because same thing. It still has to go in opposite directions of the original, but you can only add onto the 3' side. So as you are unzipping this way, the bases have to get added on the other direction. And so we do that, but it's a little trickier, because we unzip to the left, but we have to add bases to the right. So what it ends up doing is creating small little sections that go in reverse. That it will start here, and it will make a little snippet, and it will go backwards. So it just makes these little tiny segments. It will start here, and then go backwards, always adding onto that 3' side. So you end up with these little fragments of DNA. They're called Okazaki fragments. And then there's more enzymes that link them all together. I have so many Big Bang Theory memes, but I like the helicase. So this is showing that DNA replication. I need my pointer. I have my pointer today. And the issue we have with the fact that it's double-stranded and anti-parallel. So on that first strand that it started with the 3', just like mine, we have that DNA polymerase, and it can only add onto the 3' side of that new strand. So that top strand, not an issue, because as you unzip to the left, it just keeps adding on new bases, matching up T's to A's and C's to G's. And so it's called the leading strand because it's not an issue. Unzip to the left, add to the left. But it's this lagging strand, because it takes longer, that we run into that issue, because the new strand has to go in reverse, adding only to the 3' side. We make these little fragments called Okazaki fragments that we lay this little red primer down, and then we go in reverse. And then we'll lay another little red primer down, and then we go in reverse. And then we have other enzymes that clean everything up and put it all together. But it takes a little bit longer, so we call it the lagging strand. So when we make new DNA, one strand is easily copied, because you just unzip and then add new bases. But the other strand, that lagging strand, just has to do everything in reverse and just has to do one snippet at a time. I think I've got a video that shows this. Ignore the background audio. Okay, so I tested my audio. What do I have plugged in that's now making issues with my audio? Unzipping. Stabilizing protein.
One DNA strand encodes the leading prime to 3' 2. It forms in pieces called Okazaki fragments. Lays down new DNA. The process repeats.
Other things that clean up everything.
Primers with DNA.
DNA. Again, anytime a cell replicates, obviously it's going to have to make all new organelles. It's going to have to make more DNA as well. So we have to have DNA replication when we make new cells. Now, as I mentioned before, there are genes that control various features, hair color, eye color, whatever. We've got lots of things that control our features. That's all based on little tiny genes. Genotype is just talking about the genes themselves and what they control, because it's got the word gene in it, versus a phenotype. The gene is the actual gene and what it controls. The phenotype is what does it look like? It's what is the physical characteristic that we can see. Genotype, this is the actual gene. Phenotype is the actual characteristic. You have a gene that controls your hair color. You have a gene that controls your eye color. Your genotype controls your phenotype, which is the eye color. I always think the phenotype is your physical characteristic, something you can see. Now, I put a little note that genotype does not always equal your phenotype. You have a gene that controls your eye color, you have a gene that controls your hair color, you got a gene that controls just about everything. But I know there are more than one gene that controls your hair color. There is more than one gene that controls your eye color, which is why you can have like 100 different shades of blonde or 100 different shades of brown, and you can have like every shade of blue or hazel or whatever color for eye colors. Some genes, although you have genes that may control something, doesn't mean they're turned on. Like depending on your past history, maybe you've got someone in your family that's got red hair and you're like, oh, I don't have red hair. Doesn't mean you don't have a gene in your body right now that says you would have red hair. It's just not always turned on. You have genes that are not always active. So that's why genotype does not always control the phenotype, because some genes are not turned on or not active. Now, how do we get information from the genes, those snippets of DNA, to actually code for something? And it's a two-step process known as transcription and translation. We get that information from our DNA, those genes, to actually translate into something. Now, it's called the central dogma of genetics, meaning it's an extremely important. The fact that we have DNA, and through transcription, we can make RNA, specifically messenger RNA. And then from that RNA, we can translate it into polypeptides or proteins. So the idea is that we're going to take DNA and code it out into proteins. Why proteins are so important. All your enzymes are proteins. Proteins are structural material, building material. We have proteins everywhere that can pretty much control everything. And it's all because of our DNA. Now, before I get it, because I can't remember exactly what my next slide is, a little bit of notes on just, differences between DNA and translation, or the DNA transcription going from RNA and the translation. One, in a eukaryotic cell, where is DNA found? In the nucleus. DNA can never leave the nucleus. Just so you know. DNA can never, ever, ever, ever leave the nucleus. Now, if you're a prokaryotic cell, you don't have a nucleus. It's just the nucleoid region. But DNA can't leave the nucleus. So we have to make a copy of it. I liken this to kind of like my story on if you went to the library and there's a super old, super important book. It's like the book of life. It like will explain everything to you. But there's only one copy and it cannot leave the library. But you're like, but I need to know how to do whatever. You can go to the library, open that book up, and make a copy of a page. No problem. So the DNA is that book. It's the book of life. It contains all the super important information, and it has to stay protected in the library. So it cannot leave the nucleus. But we can still make copy of pages, meaning I just want to know a little bit. I just need to know, something from page 87. I can make a copy of just that one page. And that's what transcription is. It's not making an entire copy of the entire DNA, but it's making a copy of just a gene, just one little snippet of information that controls just one thing. And because the DNA is in the nucleus, this is where transcription is going to take place. It's going to make just a copy of a small snippet of DNA inside the nucleus. And that copy is now called RNA. It's not the exact original. It's like it's not the exact original straight out of the book, but it's a copy of it, and it's called RNA. RNA can leave the nucleus just like that copy can leave the library. Now, from there, you made a copy of that page. It has all the information you need, but it's in a completely foreign language. And so your next step is to take that information on that page and translate it into something usable. That's why it's called translation. So in here, it's translating it from a language of RNA to the language of proteins. A polypeptide, poly just means many, just means putting a lot of amino acids together. It's many amino acids. And a lot of amino acids is a protein. We first just make a copy of the information because DNA can't lose the nucleus. Then we have to translate it into something usable, the proteins. Oh, I was going to say, this is your basic central dogma of genetics, is you take your double-stranded DNA, and I want to make a copy of just one small snippet. So like, there would be way more DNA than just that little snippet right there, but I'm going to make a copy, and I'm only going to copy one strand. DNA is double-stranded. I'm only going to copy one strand. And so I'm going to copy this bottom strand, and if it started with a T, I'm going to bring in an A. Now, the difference here, though, is that this is now RNA. It's not the exact original as DNA, it's just a copy, and it's not the exact. And RNA does not have T's. It has something similar in structure called a U or uracil. And so anywhere a T would get brought in, a U is going to get brought in instead. So if there was a T, it brings in an A. If it has an A, it brings in a U. If there was a C, we bring in a G it brings in a C, and so on, until we've got that copy. That is going to be one of the very first things you guys do on that worksheet I handed out, is I gave you a snippet of DNA. I just gave you the one strand, and the first thing you have to do is write in that RNA code. That if it had a T, you bring in an A, if it had an A, you bring in a U, and so on. Translation is taking that information and reading it and translating it into the language of amino acids. And a long chain of amino acids is called a polypeptide. That's like your basics, but we're going to break it down before we get there. So transcription. The information in DNA is copied as RNA. The things that are helping do that, there is an enzyme called RNA polymerase. It's an enzyme enzymase. And it brings in the RNA nucleotides. So if there was the T, it brings in the A. If there was an A, it brings in a U, because it's RNA. And so on all the way through. So we just have a new enzyme, RNA polymerase, that's bringing in the A's, U's, C's, and G's, and then we have the nucleotides themselves. Or a couple different RNAs, I don't care, you know them. Let's see if I can switch back, because we're going to watch this happen. I love this video. I'll get rid of this. Couple things just to identify. This pink thing, I'm going to turn the lights off. The pink thing is your double-stranded helix. So one of the very first things that we're going to have to do, again, this is all going to be happening inside the nucleus, is we do still have to yank it apart so that we can still match up the correct A's, U's, C's, and G's.
It's the
A gene, again, it's just a snippet of DNA that codes for something.
DNA called RNA.
So then it's going to do translation, but we're not there yet. Like it, because it is happening in real time. That big blob that was unzipping it and bringing in the letters, that was just the polymerase that's unzipping it and bringing in the A's, U's, C's, and G's. So again, we now have our new RNA. We call it messenger RNA. It's carrying the message from whatever page it is that you copied. But we now have to translate it into the language of amino acids or the language of proteins. And so our participants in translation, we have our messenger RNA. We have ribosomes, because if you remember, what is the function of a ribosome? Make what? Proteins. Proteins in translation, ribosomes are the location of where all of this is going to happen. So just like you had transcription happening in the nucleus, well, translation is happening in the ribosomes. It's meant to make proteins. And you have another molecule called transfer RNA. So messenger RNA, that carries the actual message itself, but you have a molecule called transfer RNA that's actually going to help bring in the correct amino acids. Now, when we read messenger RNA, we read it 3 letters at a time. They're called codons. So we would read the first 3 letters, AUG, and that would code for this amino acid. The next 3 letters, the CGG, would code for this amino acid. The UAC would code for this amino acid. The UUA would code for this amino acid, and so on. We always read it 3 letters at a time, going from RNA to the amino acid. Transfer RNA molecule is the one that knows which amino acid to bring in. So it is the molecule that's going to bring in the methionine that knows to recognize AUG. How it knows to recognize AUG is that on the structure of this transfer RNA, I always think it kind of looks like a T, is that it has a 3-letter code that's the opposite of the codon it's reading. And so it's called anticodon. So codon is always the 3 letters of RNA. And this has 3 letters that are opposite of it. Opposites attract. So it's kind of like, oh, you're my opposite? I need to bring in the amino acid that I carry. So if there's a transfer RNA that's reading AUG, it would have UAC directly on the molecule. That UAC recognizes that, and then it drops off the methionine. So the transfer RNA is reading it 3 letters at a time and then bringing and dropping off that amino acid. So it's transfer RNA because it's transferring the amino acids. It's bringing them in. Is in your ebook because you're going to need it for your worksheet. It's figure 9.15 if you want to I don't remember the page number. I didn't write it down, I don't think. But if you're trying to find it in your ebook, because you're going to need it, these are the possible codons. So you have A's, U's, C's, and G's. There's 4 different nucleotides in RNA, and there are 64 different ways to mix those up into 3-letter sequences. So 64 different ways you can mix around A, U, C's, and G's into 3-letter codons. And this is showing you what each 3-letter codon, what amino acid it codes for. And so GCU codes for alanine, ACU codes for threonine, CCCU codes for proline. But if you notice, there are 64 different ways you can mix these different amino acids or these different bases around into codons. But how many amino acids are there in the human body? There are 20. The human body has 20 different amino acids, but there are 64 different codons. There are some prokaryotic cells that have more than 20, but we're just going with 20 because it's humans. 20 different amino acids, 64 different possible ways to mix these around, which is why, yes, there are more than one codon that can code for the same amino acid. So again, 64 different codons, only 20 different amino acid choices. So that's why you see some of the same codons code for the same amino acid. But there's also a couple color things up there to make note of. The first AUG is always where the ribosome knows where to start reading. The ribosome, its job is to do translation and make that protein. It always is going to recognize AUG. And anytime it sees the AUG, it's going to bring in the very first amino acid, which is methionine or MET. So that AUG is generally called the start codon. And so that's why even in here, my very first 3 letters are AUG. That's where it's always going to start, because the ribosome has to know where to start. There's also 3 different codons up there, so 3 possible combinations of your A's, U's, C's, and G's that don't code for any amino acid. Ignore this, because this is a very specific bacteria. But there are 3 different amino acids that don't code for any amino acid whatsoever, which means that once a ribosome encounters one of those 3 codons, it just stops translation. It's like, okay, I'm done making my protein. And it is. It means it's made it exactly correct once it gets to one of those stop codons. Now, just a little bit, since the ribosome is reading this message, a couple little things on the structure of ribosome. Ribosomes are actually structurally 2 blobs. I don't know how else to describe them. They are 2 blobs, and those 2 blobs will come together and read that message. Now, whether it's a ribosome found in a prokaryote or a ribosome found in a eukaryote, size-wise they're a little different. Function, they are not. So they're still made up of 2 blobs that are going to come together to form the full ribosome and start translating. You know, again, whether it's eukaryotic ribosome or prokaryotic, their size is different, their function is not. There still are 2 blobs, because you're going to see that in that continuation of the video. 2 blobs are going to come together around that messenger RNA, that yellow chain that it made, and it will translate it into the language of amino acids. So here's the 2 blobs. This big orange thing, that is your ribosome. The 2 blobs come together along that message, and then it goes through, it's known as a 3-step process: initiation, elongation, termination. Initiation, your ribosome, the 2 blobs are going to come together, Elongation, you have that transfer RNA that is reading the 3 letters at a time and bringing in the correct amino acids. These are just the 3-letter abbreviations. So it's elongating. It's just traveling along, ribosome's traveling along as it pulls that messenger RNA through, reading it 3 letters at a time, bringing in all those different amino acids and stringing them together. And it will continue to do this until it encounters one of those 3 stop codons. At that point, there's no amino acid to bring in, and it's done, and it's made that new chain of amino acids, which is going to be the protein. So it's your initiation, elongation, termination. Now, inside the ribosome, there are 3 spots, because the ribosome's not very big. There are only 3 spots that really hold about 9 different bases in here, and they call each of the 3 spots, where it holds 3 bases each, an A, a P, and an E. So as messenger RNA comes in the ribosome itself, it's right away where the transfer RNA is going to start reading it. So I always think A is for a protein. It's just entering and it's starting to get read. The P is for polypeptide. It's where it's stringing amino acids together. And so it's the little polypeptide bond that it forms there. And E is for exit. You know, these things are now going to exit the ribosome. It spells ape, but backwards. Again, this is just showing how you can have your ribosome, 2 little blobs coming together, and your messenger RNA, and it's reading it 3 letters at a time in your A site where it starts, the P is where it puts it together, and the E is where that new amino acid is leaving. Now, translation has to happen. It has to happen in all of the cells in our body, and it has to turn out tons and tons and tons and tons and tons of protein. When I say tons and tons, we're talking trillions of proteins, like every second right now is happening in your body. To help speed that up, you can have ribosome reading the same gene at the exact same time. This is an actual picture of a messenger RNA sequence and lots of ribosomes all attached to it and all making these new polypeptide or protein chains. Speeds it all up. You don't want just one message, one ribosome, one protein. It's like, let's have a bunch of ribosomes. Let's have a bunch of amino acid chains or proteins getting made at the exact same time. Now, same video, but I'm going to speed up to where we left off. Switch my audio. We left off that messenger RNA snaking away from the nucleus. And what, I'm going to turn the lights off. What organelle is it going to go find? Makes proteins. It's going to go find itself a ribosome. And again, you'll see the 2 blobs come together.
Or the ribosome.
There's the 2 blobs.
It translates the genetic RNA into a string of amino
It's your transfer RNA.
The amino acids are the small red tips.
Trillion molecules of that per second. You think about how many you've made just while you sat in here today. Again, it just depends on what you are pulling through the ribosome. The ribosome could care less about what it's reading. Now, whether it's a prokaryotic cell, whether it's a eukaryotic cell, same basic processes. DNA in a prokaryotic cell, again, it's circular, whereas in a eukaryotic cell, it's linear. In a prokaryotic cell, there is no nucleus, so it doesn't have to worry about doing transcription in the nucleus and that messenger RNA leaving. Whereas in a eukaryotic cell, all of that reading of one strand, making a copy of the one strand, that transcription and making your messenger RNA, all has to happen inside the nucleus, and that messenger RNA then leaves. It's still going to get read. You still have that message, and it's still going to get read by ribosomes, the two blobs that are going to sit there and read it 3 letters at a time and start making your amino acid sequence. A nascent protein just means it's like a building protein. It's still getting bigger as it's adding one amino acid at a time. Now, you guys have the worksheets. I'm just going to get you started on the worksheet. And my note, we are going to talk next class on the mutations, so don't even worry about that yet. Otherwise, other than the mutation part of the very first part, pretty much we're going to start it, and you should be able to get down to and answer like all of this top part except the very bottom, because we're going to talk about bacterial conjugation next class. But I give you the DNA sequence. Now, a little bit at the very top, I wrote it down somewhere, I forgot it. It says complete the mutation exercise, follow the instruction. And it says these DNA sequences are just the beginning bases of a gene that is involved in lactose catabolism. Meaning you're building or going to be making the protein called lactase. It's an enzyme, it's a protein that's used in helping break down lactose. This is only the beginning, because my note, it will not have a stop codon at the end. And everyone's like, there's no stop codon. I know, because I think I wrote it down. The actual nucleotide sequence to make the lactase enzyme is like 14,000 bases. I give you like 20. So that's why there's no actual stop codon. There is, 14,000 bases later. But I give you the DNA sequence, then you have to create the RNA sequence. That's the transcription. And then from that, write in the amino acids that would then get brought in. Now, I've got 4 strands here. The very first one is known as the wild type strand of DNA. Wild type, as crazy as the word itself, wild means normal. It's like the normal DNA that's found out in the wild, meaning there's no change to it. It is the normal. I don't know why they call it wild. Strain A, B, and C, my little note, I changed the DNA just a little bit. And we'll talk on Wednesday about mutations. I created a mutation in strain A, B, and C, and then you'll have to answer some questions based on the mutation, so I guess you can't answer A or B at the bottom. You're going to have to answer the questions, based on the mutations that I created, could this strand still make that enzyme lactase? Could it still do its job based on the mutation I gave, and why? But if I gave you the first 3 letters, I'm just going to write some of them up. I'm not going to write the whole strand up. E, A, C, T, A. All right, those are the first 9 letters. And a little helpful hint, especially once you write out the next strand, it wouldn't be a bad idea to put like a comma or a line between every 3 letters. It's crazy how many students create their own mutation by reading it incorrectly. So I even kind of spaced a little bit out. So if the first 3 letters are TAC, and this is your DNA, we're going to make the RNA strand. So in RNA, if this is a T, what's the letter that's going to get brought in? An A. And if this is an A, what letter will get brought in? A U. Again, it can't be T because it's RNA and not DNA. If this is a C, what gets brought in? G. What gets brought in with this A? U. A. U. And so again, you're going to have to go through that whole thing. But I at least want to get you started. And I want to get you started on how you go from the RNA to the amino acids. using this figure. Again, figure 9.15 in your book. So we're reading it 3 letters at a time, and apparently I can't write in a straight line. But the first 3 letters. So these are set up that on the left-hand side of the figure, look up here, left-hand side of the figure is the very first of the 3 letters. So your choices are U, C, A, or G. My very first letter in my codon is A. So I know right away I'm going to be in this row. Now, across the top is what is space in your codon. Mine is U. So out of your 4 choices, I'm going to be in this column. So I'm in the 3rd row, but I'm in the 1st column, which narrows it down to this box. And then it's read over on the far right-hand side. Here's your 3 or your 4 possible options for what is your 3rd letter. And mine was a G, so I'm, 3rd row, 1st column, very bottom, and there's your AUG. So that's how it's set up. Now, the very first codon that gets brought in is methionine. You can shorten it to the 3 letters, or you're going to have to write really tiny, to MET, M-E-T. My next codon is UAU. So the very first letter, U. The second letter, A, so right away I'm in this box. And my 3rd letter is U. And so the amino acid that gets brought in is called tyrosine, or TYR. Your 3rd codon is GGA. Don't say it out loud yet, but see if you can find it. And then tell me what it codes for. GGA. Don't say it. Got it? What amino acid does it code for? I'm going to say the glycine, the GLY. So your GGA way over here, and again, you can just write the gly. Or EU off on the worksheet. Again, that's about all you can get on there. On Wednesday, we're going to talk about the different mutations that can happen, why some of them aren't so bad, why some of them are catastrophic. And if you also wanted to work on the backside, you could, because I'm not going to lecture on chapter 10. So I do not lecture on chapter 10. And so I have you go into chapter 10, because chapter 10 is all the ways we can manipulate DNA and all the ways we can kind of play with DNA. It is really cool stuff. I wish I had the time to lecture on it, but it includes the things on, how do we use DNA and the fact we all have unique codes to identify perpetrators. How can we match up DNA to know who's the actual person at the crime scene. So in chapter 10, so the back side of the worksheet is go to chapter 10 in your ebook, the end of chapter 10 in the ebook, to the critical thinking questions. There are multiple groups of questions at the end of chapter 10. I can't remember what they're all called, but find the one that starts with critical thinking. In your ebook, it's at the bottom, page 332, and goes to the top of page 33 on the ebook. And choose any 3 of those 10 questions and answer them. Now, I think the first couple questions have like 1A to 1B. You have to answer both A and B of question 1, and that's one question. You picked question 1, and you have to answer all parts of it. But that's 9 points, so don't forget to do that. It's 3 points for each of the 3 questions that you choose for chapter 10. I don't care which ones. Just go to the critical thinking. My also little note, we started this, so I don't think you guys are going to go wrong on it. For this, don't Google this worksheet. I'll know if you do. I feel honored that someone at some point scanned my worksheet and put it on Google, but they did it incorrectly. And so sometimes I laugh a little bit inside because I've got an online section of micro right now, and this was just due this weekend, and I was like, I know exactly who Googled it because it was done incorrectly. And I'm like, I'm honored that someone put it on there, and I don't know why they put it on there incorrectly. I didn't do it. Don't try to Google it, because I've changed this worksheet so many times, and it was still done completely incorrect. So you guys know how to do it. We started you off with the transcription translation. So you can start it, you can finish those codes. Just don't answer the questions on the bottom of that first page yet, because we'll get into that stuff on Wednesday. Before I see you next class, make sure if you haven't already done the immune system exam, that's due tonight. And if you haven't done the chapter 16, the immune disorders, that's due Friday. Otherwise, there isn't any other quiz on this yet due. Mm-hmm. Let me look. Yep. You're good. Thank you.